name's Martin Kashara, and this is the University of Wolverhampton, and we are standing in the fantastic Rosalind Franklin Science Building. You are about to witness the £25 million investment in science teaching the university has made, and we're here today to actually meet a very special scientist. But that scientist is all the way up on the very 5th floor of this building. So what I thought we'd do at least, is just have a little tour around the building on our way up there before we meet the fantastic Professor Izabella Radecka. And here we are on the very first floor of the Rosalind Franklin Building. The Rosalind Franklin Building is actually divided into subject areas depending on what sort of floor you're on. So here we are on floor 1. Now, on the first floor, what we have is chemistry. So behind me, you can see that this laboratory is set up for a practical session, ready to run face to face, even in spite of our COVID pandemic, about a subject in chemistry. And what we're going to do is go up to the next floor, where actually we'll see where some of the more biological practicals actually happen. Now here we are on the 2nd floor of the Rosalind Franklin Building. Now if you remember what it looked like before, on the 1st floor, which was primarily involved in things like chemistry, actually now on the 2nd floor we're involved more in biological sciences. And here on the 2nd floor we do things like biomedical science, haematology, anything really, microbiology, anything to do with the things that infect a person or the fluids of a person. We teach people how to analyse those sorts of samples to be able to actually diagnose disease in real patients. And you can actually see behind me that the laboratory is set up in lots of different ways. You can see we've got microscopes behind us and so on. And also you might see that some of the desks have got tablet devices on a sort of arm. And actually our students, we can actually get 90 students in here at a time, when we're not in a lockdown, of course. So students get a tablet between two, and that allows them to record their data in real time without paper straight into their sort of student learning environment, which is absolutely fantastic. So we have people from all over Europe come and actually see the sorts of things that we're doing here in the Rosalind Franklin Building, because it is considered best practice for science education. But this isn't all, because we're just on the 2nd floor and we've got 3 floors left to go. So here we are on the 3rd floor of the Rosalind Franklin Building now. Now, the 1st floor, as I said, was to do with things like chemistry. The 2nd floor was to do with the biological sciences that concerned the fluids of the body and the infectious things of the human body itself. But this floor is about biochemistry and molecular biology, about DNA and about analysis of fluids that allow us to diagnose disease. And I'm talking quietly because over here we can't show you that at the moment. Right now there's actually a class happening face to face, even though we are under COVID regulations. We can still have our face-to-face practicals in a COVID-safe manner in these laboratories. And these students are experiencing how to actually use scientific equipment that is going to be their tools of life as scientists as they move forwards through their career. What we're going to do, we're going to move up to the other floors, and they are very special places indeed, because they concern the research that the university does through the Faculty of Science and Engineering. Okay, so let's go and have a look at the 4th floor. See you later, everyone. Bye. Now here we are on the 4th floor of the Rosalind Franklin Building, and this is the absolute hive of the research effort in the Faculty of Science and Engineering. Here there are many different subject areas represented, and there are postgraduate students carrying out their PhD sort of research programs, and also academic researchers here working on loads of different kinds of projects. One of the things we do particularly in this facility here is look at new and improved ways to treat cancer. And that's everything from bowel cancer to brain cancer. And every day, new discoveries are being made just in this room. I mean, how fantastic is that? But the scientist that we're going to see doesn't live here. She's one more floor upwards. So let's go and see if Professor Radecka is available on the 5th floor. Here we are on the very 5th floor of the Rosalind Franklin building. This is the top research floor of the building itself. And here we've got some fantastic facilities. On one side, we've got greenhouses that look out towards the sky and they're lit all the time. So if you look across Wolverhampton at night time, you can see a sort of pinky purple glow from the top of this building. And this is our greenhouses being lit 24 hours a day to grow the experimental crops that they're looking at here at the University of Wolverhampton. But behind me is the home of one of our fantastic research groups, and that's the biopolymers research group. And the lead of that is Professor Izabella Radecka. So let's go and meet her now. Yeah.
Hello, I'm Professor Izabella Radecka, and this is my colleague,
Dr. Johnston.
And we are in our fermentation lab in the Rosalind Franklin building at the University of Wolverhampton. And this lab is a fermentation lab. As you can see behind us, we have a lot of fermentation vessels, and this allows us to carry on our research in an area of biodegradable polymers. So we use microorganisms and we grow them most of the time using waste material, and we produce biodegradable polymers. Well, we are in the fermentation lab, so fermentation biotechnology lab. As you can see, we have fermentation vessels here. Those vessels are used to grow microorganisms. They're like little houses for them, and we can really provide them with very good environment, temperature, pH, lots of food, and we can grow them in a very good quantity, and we can produce very interesting, valuable products.
Okay, so this laboratory's got lots of different uses. We can analyse different types of plastics here because we work on degrading plastics and making biodegradable plastics to replace those degraded plastics. So in the fermenter, we can grow bacteria that help to break down plastic, and then we can make biodegradable plastic. And this machine here helps us to control the conditions of the reactor, so we can control the temperature with this little belt here. So this can heat up and warm up the vessel. We can also change the pH, and we can also increase the speed of the impellers, which spin round and make the bubbles, so that the oxygen levels can go up and makes the bacteria grow more. So before we start the fermenter, we have to autoclave it in this machine. So we fill up the vessel with water, place it in the autoclave, and it gets heated up to 121 degrees for up to about half an hour, and that kills any bacteria to make sure it's completely sterile. And then we can load whatever we need to load into it, and it can grow without getting contaminated.
Okay, so what we have here is an incubator shaker. So if we want to inoculate or put bacteria into our fermentation vessels, which you've seen before, first we have to make them happy and alive and make enough what we call the starter culture. So we prepare little flasks with a particular growth medium. We put microbes in and we just keep it here overnight until the microbes will grow. We check if they're healthy, if they are not contaminated, and then this goes to a fermenter for the fermentation process. And as Brian said, for our bioplastics most of the time in this lab, because we produce biomaterials, bioplastics. Okay, as we mentioned before, in this laboratory we work with biomaterials. So we grow the microbes first in a shaker, as you've seen before, then in a fermenter, and then after that we isolate the polymer. And when we isolate the polymer, you can see that some of them, they really like little plastic materials. They behave very similar to plastic materials, but it's fully biodegradable material made by bacteria. And for some of our research, we just use a dirty cooking oil, very dirty cooking oil, which is much different to a fresh oil. And when we use this dirty cooking oil as a food for microbes, they can just produce the bioplastic, the biomaterial. Now, we need to know how good the plastic is. So one of the methods of trying to find out if this plastic is of good quality, we use this machine here, which is called gel permeation chromatography. The biopolymers are made from chains. So polymer is a big chain, and now depending how long is the chain, that reflects on the quality of our plastic. So we have to measure the length of the chain. So we use this machine, which is called gel permeation chromatography, to assess this, what we call a molecular weight, the length of the chains in the biomaterials. So Brian, you can say now.
Yeah. Okay. So to prepare those samples, we take a small amount and we melt it down in chloroform, and then it gets loaded into the machine through the cassette deck here. So the sample goes in there, then a tiny syringe goes into the sample, sucks up a sample, and then it passes through this column here. And depending on how long it takes going through the column, we can work out its molecular mass. So on the computer will do a graph and it calculates the molecular mass of those samples. So this is our 3D printer. So we can use our biodegradable polymers, mix it with PLA, polylactic acid, and we can make 3D printer ribbons like this and create shapes like this, or even shapes like human ears. And we've actually been able to, we had a paper a few years ago where we grew human cells on these graphs as well. So it's able to be used for lots of different applications. And the fermenter can also be used to make conditions for human tissue growth on these platforms as well.
Wow, wasn't that absolutely amazing? The work they are doing in this room is literally saving the planet. It's not only replacing plastics that we can't degrade with plastics we can degrade, made from waste that we throw away every day. It's also helping to build the scaffolds that in the future we'll be able to actually grow human organs on. And I think that is absolutely fantastic. I hope you've enjoyed having a little tour around the Rosalind Franklin building today and meeting one of our fantastic researchers. So hopefully we'll see you again soon. Bye bye.